I'm Ryan Pettit-Mee, a doctoral candidate in the exercise physiology program within the Department of Nutrition and Exercise Physiology. My advisors are Dr. Jill Canali and Dr. Jean Padilla, and I will summarize my dissertation titled The Role of Heat Shock Protein 72 in Endothelial Insulin Resistance in Type 2 Diabetes, using passive heat therapy to improve endothelial insulin responsiveness. Factors such as obesity, physical inactivity, and glucolipotoxicity can increase the intracellular activity of stress kinases such as JNK. These kinases promote insulin resistance by disrupting the PI3K-AKT signaling pathway within metabolic tissues and endothelial cells. When this pathway is disrupted in endothelial cells, the effect of insulin to produce nitric oxide that induces vasodilation and subsequent increased blood flow that is important for glucose uptake is impaired. Furthermore, this endothelial insulin resistance contributes to higher prevalence of cardiovascular disease and mortality rates in people with type 2 diabetes. A key regulator of such stress kinases, known as heat shock protein 72, or HSP72 for short, has been found to be reduced in metabolic tissues and aorta from insulin-resistant humans and rats respectively. Importantly, increasing HSP72 via passive heating can inhibit overactive stress kinases such as JNK and restore insulin signaling. Yet the role of HSP72 in endothelial insulin resistance and endothelial insulin signaling is unknown. The overarching hypothesis of this dissertation was that HSP72 is decreased in endothelial cells in type 2 diabetes, shown in the red box, promoting impaired PI3K-AKT insulin signaling via mechanisms such as increased JNK activity. Conversely, increasing HSP72 in endothelial cells by passive heating would improve PI3K-AKT signaling, in part by decreasing JNK activity, as shown in the green box. For this dissertation, 3 specific aims were used to test this overarching hypothesis. Firstly, aim 1 sought to determine if endothelial insulin resistance in type 2 diabetes is attributed to reduced expression of HSP72, with the hypothesis being a reduced expression of HSP72 would contribute to endothelial insulin resistance in type 2 diabetes. To test the hypothesis of aim 1, 2 approaches were used. In the first approach, it was hypothesized that isolated aortas from DB mice, a mouse model of type 2 diabetes and colored in yellow, would exhibit blunted insulin-induced relaxation and reduced HSP72 relative to isolated aortas from DB+ or non-diabetic mice, which are colored in white. Despite isolated aortas from DB mice exhibiting blunted insulin-induced relaxation compared to DB+ mice, as shown in graph A, HSP72 expression was not different between DB and DB+ mice, as shown in graph B. In approach 2, it was hypothesized that subjects with type 2 diabetes, highlighted in yellow, would display impaired postprandial leg blood flow and decreased HSP72 in collected endothelial cells relative to healthy controls, highlighted in white. Human subjects had leg blood flow recorded by ultrasound before and after an oral glucose load, and endothelial cells were collected from an arm vein and imaged for HSP72 and JNK activity. Although subjects with type 2 diabetes had reduced postprandial blood flow compared to healthy subjects, as shown in the bottom panel of graph E, there was no difference in endothelial HSP72 expression between groups, as shown in graph F. Aim 2 sought to determine if 7 days of passive heating improves postprandial leg blood flow with increased expression of endothelial HSP72 in subjects with type 2 diabetes. The hypothesis was that 7 days of waist-level hot water immersion of 1 hour a day would improve leg blood flow responses to an oral glucose load, i.e. endogenous insulin stimulation, and would be accompanied with increased endothelial HSP72 expression. For aim 2, the same subjects with type 2 diabetes that volunteered in aim 1 had the same measurements retaken after participating in 7 days of passive heating. This heating intervention was accomplished by waist-level immersion in 40.5 degrees Celsius water for 1 hour a day. Although there was an improvement in postprandial leg blood flow after the 7 days of heating, as shown by the red data points in the bottom panel of graph E, there was not an increase in endothelial HSP72 as shown in graph F. Aim 3 sought to determine if modulation of HSP72 impacts endothelial insulin signaling and cell culture. The hypothesis was that downregulating HSP72 would impair insulin signaling, whereas upregulating HSP72 would enhance insulin signaling in human skeletal muscle microvascular endothelial cells known as SMECs. Several experiments were performed to test the hypothesis for aim 3. In the first experiment, cells were treated with scrambled siRNA as control or HSP72 siRNA to reduce HSP72 expression. After a double transfection protocol, cells were treated with or without insulin and then examined for HSP72, JNK activity, and insulin-stimulated AKT activity, which was a marker for endothelial insulin signaling. Overall, siRNA knockdown of HSP72, as shown in yellow in graph A, did not suppress insulin-stimulated AKT activity, as shown in graph C, nor was JNK activity altered as shown in graph B. In the second experiment, cells were exposed to 1 hour sham treatment at 37 degrees Celsius or heat treatment at 40.5 degrees Celsius over 5 days. 24 hours after the last treatment session, cells were stimulated with or without insulin. Overall, increasing HSP72 via 1 hour of heating over 5 days, as shown in graph E, did not enhance insulin-stimulated AKT activity as shown in graph G, nor was JNK activity affected as shown in graph F. In the third experiment, cells were exposed for 24 hours to either a vehicle solution that served as a control or to a glucolipotoxic milieu containing high glucose and palmitic acid. After this exposure, cells were then stimulated with or without insulin. The glucolipotoxic milieu was to mimic high glucose and high fatty acid conditions experienced in type 2 diabetes. Overall, 24-hour exposure to a glycolipotoxic milieu suppressed HSP72 as shown in graph I and overall AKT activity as shown in graph K, but did not influence insulin-induced AKT activation as no interaction was found that is also displayed in graph K. Again, JNK activity was not altered as shown in graph J. In the last experiment, glucolipotoxic cells were exposed to 1 hour of sham treatment at 37 degrees Celsius or heat treatment at 40.5 degrees Celsius. 24 hours after the treatment session, cells were stimulated with or without insulin. Overall, increasing HSP72 via 1 hour heating as shown in graph M did not improve insulin-stimulated AKT activity in glucolipotoxic treated cells as shown in graph O. Furthermore, JNK activity was not affected as shown in graph N. In summary, counter to the hypothesis of AIM-1, endothelial insulin resistance in type 2 diabetes mice and humans was not associated with reduced HSP72 in aortas and endothelial cells respectively. Counter to the hypothesis of AIM-2, improved postprandial leg blood flow in subjects with type 2 diabetes following 7 days of passive heating was not paralleled with increased endothelial HSP72 expression. Counter to the hypothesis of AIM-3, downregulation via siRNA knockdown or upregulation of HSP72 via heating did not impair or enhance insulin signaling as demonstrated by insulin-stimulated AKT activity, nor was JNK activity altered. Based on the accumulated data, this dissertation concludes, firstly, HSP72 is not a key determinant of endothelial insulin resistance in type 2 diabetes. Secondly, passive heating may be an effective treatment for improving postprandial blood flow in subjects with type 2 diabetes by a mechanism independent of HSP72.